To suppose that the eye, with all its inimitable contrivances for adjusting the focus to different distances, for admitting different amounts of light, and for the correction of spherical and chromatic aberration, could have been formed by natural selection seems, I freely confess, absurd in the highest degree. Charles Darwin. My name is Ulysses Velasquez, and I will be doing my presentation on the evolution of the eye. I'm especially fascinated with this subject because I am an aspiring optometrist, and I was intrigued by the idea of the evolution of the human eye. I want to begin with my thesis statement. Can we use extant species to determine the evolution of the human eye from simple photoreception to complex visual processing? First, I would like to do a brief overview of eye anatomy so we're familiar with the terms. We'll begin with photoreceptors, then the retina. We'll talk about the iris and the pupil, the crystalline lens, also just known as the lens, and finally, the cornea. And these are building on each other in succession. First, let's talk about the photoreceptor. In the human eye, there are two specific types of photoreceptors. The first are called cones. These are used for color vision and fine visual resolution. Within the retina, there is a fine concentration of cones in what is called the fovea centralis. This dense area of cones is responsible for our fine vision when looking directly at something. Rods are a second type of photoreceptor. These are responsible for phototransduction of light and are used when we are in the dark. We find a greater concentration of these in the periphery of the retina. Next, we have a model of the human eye, and I will begin at the cornea and work backwards. First, we have the cornea. It's this front surface of the eye associated with refraction. When light hits the cornea, it bends the light. The cornea makes up 3/4 of our optical power. It's very important to fine, detailed vision. As light hits, it diffracts off the cornea and passes through the pupil, which is just a dark hole. The size of the pupil is determined by the iris, which changes. We know this as dilation and constriction. As the light hits the lens next, it is further refracted or bent, and that is the main purpose of the lens, the last part of the refractive power of the eye. The light falls on the back of the eye, on the macula, specifically on the fovea centralis of the macula. This makes up our fine vision. Other light will of course hit other parts of the retina and will be received. The message then is transferred through nerve endings into the optic nerve and passes to the back of our brains. Next, I want to talk about the species that we use. First, we'll use the euglenid, then the mollusk, nautilus, the box jellyfish, and finally we'll end with the most complex eye, the octopus, and we'll compare it to our own. First, we have the euglenid. These are single-celled organisms. They use chlorophyll to produce energy. They're very important when studying the evolution of the eye because all the euglenid can do is determine if there's light or not. That is the most simple form of light perception. Is there light or is there no light perception? The euglenid, at the very end of its flagellum here, has a receptor that helps it to understand where the light is located to move towards the light for photosynthesis, for its survival. From here, I have done a little bit of cartooning to help us understand the process of the evolution of vision. Here we have a simple photoreceptor, like that on the euglenid. If you were to take many photoreceptors now and put them together, you'll have something like a retina. If it's flat like this, you'll still only be able to determine if there's light or no light. But with a simple folding or bending in the retina, you get this cupped shape. The cupped shape allows us to understand what direction things are coming from, and it's very important in the evolution of the eye. So let's look at a species that has this type of eye. The eye cup of the mollusk. Folding of the flat retina gives birth to directionality. As we can see here, as a dolphin passes in front of the eye cup, the many parts of it will land on the cup at different times due to some parts being closer and some parts being further. This allows the mollusk to know in what direction something's moving. Here is a simple view of what that would look like. There isn't very much detail, but what this does is it opens a new realm of possibilities. Now, a species can understand if a predator is moving in its direction or not, from side to side. And prey can also know if its food source has moved past it. This is very simple processing, and some scientists believe it is the first form of true vision. From here, we move on to the cupped retina and its further folding. This is yet another cartooning I've done. As it continues to bend downwards, we see that it forms a pinpoint area here. This is known as a pinhole, and you can do this with your hands. You can form a little pinhole to focus light through it so you can see better. This type of visual processing allows for even a finer detailed vision and directionality and light perception. One of the most famous species that has the pinhole apparatus is the nautilus. And these are a beautiful species that you can find in the water. They're very majestic. The pinhole of its eye lies right at the center, almost like a pupil. So now not only do we have a retina in the back, but we also have the formation of a pupil to focus the light onto the back of the retina. Some scientists believe that the folding and the addition of a pupil protects the retina from large objects coming towards it and hitting it. Damage to the photoreceptors would make vision very difficult, and it further increases likelihood of survival if you have a predator, as you can see where they're coming from. From here, we move on to even more complexity. Not only do we have the pinhole, but now we have the introduction of the lens. Some scientists believe that the lens, suspended by these zonual fibers, was added to the eye to protect it from further material coming in and damaging the retina. This lens almost acts like a protective shield. It's clear because if it was opaque, then you would not be able to have vision, as the light would be absorbed into the lens. So it is clear so that light can freely pass through it, but it's sturdy so that material cannot pass and damage the retina. A species that demonstrates this is the box jellyfish eye, which at first sight looks like something out of a movie. Here in the cartoon or drawing, we see the lens here, free and open space. Behind it, the retina, protected by this large lens. Box jellyfish actually have two eyes, and both eyes work in the same manner, having a lens and a retina. So, as light comes in, it passes through the pinhole, through the lens, and onto the retina. From here, yet again, more complexity. We have our retina, our pinhole, which acts like an iris. We have our lens, and now we have the addition of the cornea, which is also thought to be further protection against foreign objects coming in and damaging the lens, as it is now an important attribute of vision as it refracts light onto the retina. With the addition of the cornea, we have even finer detailed vision. This is like the vision that you and I have when we are looking at something. Very fine detail. And we share this attribute with the eye of an octopus. Very amazing. So, here we have the eye of an octopus. Though it looks very different to our own in some manners, as the cornea does not protrude as far out as ours does, we do see a cornea, an iris, a lens, and a retina, which are all connected to form the optic nerve. The eye of the octopus has light perception, like the euglenid, directionality, like the mollusk. It's able to focus light on the retina, like the nautilus, using its pinhole. The retina is protected by a clear lens, like the box jellyfish. And now we have the addition of the cornea. And the cornea is something that we have on our eyes. And like I said, it makes up 3/4 of our optical power. And it's a very important part of vision. In conclusion, we see that it is possible to use extant species to hypothesize the formation of the complex human eye. We know that fossil records make it very difficult, as things that are not like bone have the tendency to decompose unless they're flash frozen or kept in amber like we've seen in other fossil records. Some questions that I have are, how might we use species on the land to do the same demonstration, as I've used mostly amphibious species? And furthermore, what is the next step in the evolution of the eye? There's amazing research being done currently of contact lenses that will allow us the ability to zoom in on objects, and also contact lenses that can give us night vision. There are artificial lenses like the lens we saw in the demonstration, that can give people 20/20 vision even in old age. So there's so much more that is coming as far as the evolution of the eye goes and the evolution of our technology. I'd like to also mention that within each figure in the PowerPoint, you can also see a reference of where to find that image. I'd like to also mention and acknowledge the work of Nilsson, as he has done a lot of research in presenting the evolution of the eye, and I have used many of his research papers and trying to do my own. And I'm so thankful for the opportunity to discuss this amazing organ that we have in the human body, the eye, that gives us the opportunity to not only to see things in the world around us, but to see our family, to see our friends, to see in color, and to perceive the world in a way that couldn't be imaginable were we lacking it. Thank you for your time, and have a wonderful rest of your day.